I'd like to welcome you to our 16th annual Plant Genome Summer Student Symposium. I'd like to start by introducing our judges. We've got Leila Faiz. Leila, there's Leila, who's a postdoc here at the Boyce Thompson Institute studying aphid spit. We've got Cynthia Holland, who's also a postdoc here at the Boyce Thompson Institute studying cardiac glycoside biosynthesis. We've got Margaret Frank, who's in the back corner there, who is the newest faculty member here in plant biology and is studying grafting. And then we have Ursula Jander, who is an emeritus faculty member from Washburn University in Topeka, Kansas, and is notable because she mentored most of my early research. And I'd like to talk about that a bit, and then also talk about my own first internship as a high school student and the research I was doing, which was considerably more dangerous than any of the research that you guys are doing. So this picture I've already showed some of you as part of the lab notebook talk. One of the projects that I was mentored in was looking at pond snail heartbeat, so measuring snail heartbeat in terms of temperature and snail size, and generated some data. Another project that I was involved in is illustrated here. I'm marking honeybees and then measuring the bee dancing behavior when they came back to the hive to see how they indicate direction and distance to the food source. And that's my bedroom right there where the bees were flying to. And of course, not all mothers would let you keep a beehive in their bedroom, but mine did. And this science fair project then also prepared me for my first internship, because I developed not only a skill in marking bees, but also a good tolerance for bee stings. And this here is Sydney Cameron. She's now a professor of entomology at University of Illinois, Urbana-Champaign. When I first got to know Sydney, she was a graduate student at University of Kansas studying bumblebees. And at least according to Sydney, she had a strong allergy to bumblebee stings, and therefore needed to hire somebody to do the work of taking care of the bees where there might be stings involved. I don't know if that's true, but I was hired to do the dirty work for the bumblebees, basically. So this is what a typical bumblebee nest looks like. So we have usually one queen bee and maybe 100 or so worker bees. You've got these open cells here where they're storing honey. Some of them have eggs or small larvae in them. Some of them are covered, so those have pupae in them, so the bees that are soon to become adults. And only one queen is laying the eggs here. And so this is important for the bees, so all the other bees know that there's one queen present, and they communicate this to each other in various ways. And this is what a bumblebee nest looks like when you have it in a box with a glass cover. But in reality, what a bumblebee nest looks like is more like this. They tend to be underground and very dark. And so the bees are not communicating by seeing the queen. They're communicating by interacting with the queen. The queen sort of bosses the other bees around physically and also bosses them around chemically. So she produces a queen pheromone that lets the other bees know that there's a queen in this bumblebee nest. And if you take the queen away now, something interesting happens in that you get development of these worker bees that they start laying eggs. So here's the ovaries of a queen. This is a normal worker. And then in a nest where you've taken the queen away, some of these workers start developing ovaries. And that's where the interesting part is that some, but not all of the workers develop ovaries. So the more dominant workers develop ovaries. And so that was my job, was figuring out which ones are the dominant workers and what's changing in these dominant workers. So what that involved is then going back to this bumblebee colony. So we took out the queen, and then we had to observe the other bees to see which one is the boss bee when the queen is gone. And that, at first glance would be very difficult because to us all these bees look pretty much the same. But what you can do is you can stick a number on the back of each bee, and then you can tell which bee is which, and you can say, well, bee number 2 is bossing the others around. And as you can imagine, there's some risk involved in this process, opening up a colony with 100 bees and sticking a tag on the back of each bee. And so we would do observational studies trying to figure out which is the boss bee, and then also determine whether these bees, as they're developing this dominance, are also developing a queen-like pheromones, was the other big part of the project. Because the bees are not only physically dominating the other bees, they're also chemically dominating them. And so bees, in their communication, have a variety of odor sources on their bodies. And particularly important here is the mandibular gland, which produces the so-called queen mandibular pheromone. And so my job then was, once we figured out which one was the dominant bee in each nest, was to catch one bee, the dominant bee, hold the bee with one hand and hold a piece of filter paper with the other hand, let the bee bite into the filter paper, and then this queen mandibular pheromone would be in the filter paper, and that would be sent off for mass spectrometry. And sure enough, over time, as the bees developed information about who was the dominant or boss bee, that bee would then develop queen-like pheromones in its mandibular gland. I'm not going to be showing those data in the interest of time, but that's sort of the result of my high school internship with Sidney Cameron, was to do this kind of research with bumblebees to see what physical or chemical changes were occurring in worker bees when there's no longer a queen in this bee's nest, and specifically marking bees and then collecting the pheromones and probably getting stung about 100 times that summer. So with that, I want to continue with your talks, and just a little one procedural note. So each of you have 15 minutes for your talk. I know you've all practiced, and we'll all be taking 15 minutes, but just in case, I'll be sitting here in the front row, and if you're getting towards 12 minutes or above 12 minutes, I'll be holding up a sign that looks a lot like this. And that's an indication that it's maybe time to taper off and say it's time for questions.